Cell
to cell.
Yeah! Hey everyone, welcome back to Cells to Cell. My name is Heem, that's Nick, and today we have a very special guest with us here today. Her name is Victoria. You want to introduce yourself real quick?
Yeah, sure. Hi everyone, my name is Victoria Laney. I'm a fourth year PhD candidate in Dr. Jungrong Lu's lab, so similar to Ryan and Hong, who have also been on this podcast. Yeah.
Cool.
Awesome.
So
Nanomedicine, nanoparticles.
Yeah, that's basically what we do. That's our bread and butter.
I was going to say, that seems like a common theme on this podcast thus far. But it's a great lab to kind of introduce everything because we've talked about nanoparticles a little bit on this podcast. And I think that it's a good way to kind of start looking at how you can use nanoparticles for regenerative medicine and start pushing it into the clinical areas, start using it on humans. So what were you going to say? I'm sorry to cut you off.
Nothing. So first, let's just tell the audience about you. So your background a little bit, where you come from. So I guess we can do undergrad, what you majored in undergrad. We know you did a master's, so where'd you do your master's and what was it in? And then how'd you find case?
Yeah, sure. So I guess a bit about me. I was born in DC but grew up overseas and then moved back to the States for undergrad. I went to Hopkins and I was doing chemical and biomolecular engineering there. Took a year off and was working for like a biotech startup / contractor for the NIH. And I really liked it, but also it was a little bit chaotic. And I kind of knew that I needed more like scientific background to make it in the scientific field. So I went back for a master's, still at Hopkins, still in chemical engineering. And from there I was like, wow, this research thing is pretty cool. Mm-hmm. Let me just like keep going. So then applied to grad schools, ended up at Case.
That's awesome.
Found Dr.
That is
Lu.
really cool.
So we asked everybody this because we both finding how we found Case is so bizarre, but did you know like what Case Western was, like kind of like what they did, how successful they are at everything, or most things?
Funnily enough, I did. Also in my master's, we had a collaborator at Case.
That's really cool.
In the bio department though. And so from there, I was like, oh, Case is the university that publishes like these big papers.
Oh yeah.
So I came here for like a tour and they were like, look at all the hospitals, and everyone was super nice. And also like the culture of Case is very much like people centric. Mm-hmm. So when you're doing like clinically translatable research, everyone's there like thinking about the patient first.
Definitely.
So that's
awesome.
a nice aspect.
Yeah, that is really cool.
Yeah.
Now, Johns Hopkins is in Baltimore. Mm-hmm. How do you find that different than Cleveland? Is the food better or worse than here? Because I struggle with the food in Cleveland. I do.
Funnily enough, I'd say that they're actually pretty similar in terms of like the city itself, but the food is much better in Baltimore.
Yeah.
Baltimore's just like so close to the DMV, and the DMV has like, DC area has a lot of like international like food options. Yeah. So that's like trickled over into Baltimore too.
That's pretty cool.
Yeah. Here you have to kind of, it's sporadic, you have to drive around.
Yeah, I was gonna say, you really have to find the good spots.
Yeah.
And I think I've only been up here for like 3-4 months. And I found a couple good spots, but I come from Cincinnati, and Cincinnati's very Germanic, Italian area. So, but I've grown up there my whole life, so I know exactly where I want to go.
Yeah.
And I haven't found a good sushi place up here yet. I need to find a good sushi place up here because I knew where they were in Cincinnati. I miss my sushi. But I think that I find it interesting that everybody who comes here, whether they're from the West, from the eastern side, they come up to Cleveland and they all kind of have the same kind of thing. Like, and it's interesting how Case is here, but everybody wants to get out of Cleveland. You know what I mean? Isn't it? It's kind of bizarre, but I think it's kind of.
But everyone knows the Cleveland Clinic.
But everybody knows
the Cleveland Clinic. Like, there's a big Cleveland Clinic presence in Dubai, and you went there for a conference this summer, right?
Yeah, I mean, I went there for a vacation, and
okay.
Shortly after that went to another conference, so I just tacked them together mentally. But yeah, I didn't know about that. They have Cleveland Clinic is there, and then also NYU is there as well, but in Abu Dhabi.
Cool.
Yeah, I really liked it there. I was like looking up how I could become a professor there.
Really? No,
yeah, I was like,
Dhabi,
NYU seems like a good option.
Yeah. Was it like hot there?
Ridiculously hot. Yeah.
Dry hot?
Yeah, it was a dry heat, and also like you can't walk around because of that. It's like you'll walk like for 5 minutes and just be like drenched in sweat.
No way.
So it's a little bit
wow.
undesirable.
That sounds terrible.
But a lot of funding.
Yeah.
Good sources of funding.
There's a lot of money there.
Tons and tons. Infrastructure is beautiful.
Yeah, definitely.
So it sounds like you're pretty well traveled.
Yeah.
So you said that you were born in DC and then you went out of the States. Where was your stint out of the States?
So I moved around because my parents worked for the State Department. So it was Cameroon, Tanzania, Zimbabwe, and Australia.
Wow.
Yeah.
And Australia.
And
Yeah.
Australia, like
Because it was like around Africa, and then one time we were like, let's go to Australia.
oh my
So,
gosh.
Yeah.
Wow.
That's
pretty
wild.
cool.
So I'm sure that definitely aided, leaving and then coming back and just, I don't know, I feel like that just makes you just a well-rounded person, like both in your education, just like
Thank you.
as a people. No, it really, it does.
You get to know different cultures.
Yeah, and I
people.
think that that's so important, and I wish that more people could do it. Like, I've only been out of the country one time, and I went to England. Like, I went to London, and like
London's cool, though.
London was so cool, and there
Yeah.
are so many different cultures there and everything. But I've never been like I told you before. I want to go to Africa. I think it'd be really cool to go
Yeah.
to Africa and just see it. But my question, the build up to this question is so. How did that and seeing science around the world affect your science when you came back here to Johns Hopkins and now at Case?
Yeah, so initially what I wanted to do with my scientific career was work on things like point of care, like low resource environment type of solutions. That was my goal, and from there I ended up going into nanoparticle work because of it. Because for instance, like when they're looking at doing point of care, the majority of the work that they're doing is actually nanoparticles, and then they'll combine it with like a lateral flow assay, for instance. That's what they were trying to do with all of the like COVID tests, basically. Like there were so many nanoparticle groups who were like, this is applicable. So my, I guess, mindset coming into nanotechnology is being a little bit more resourceful and creative with some of the ways that you approach science and devices, for instance. We work with an MRI, and it's constantly breaking down, so you have to come up with some creative solutions to keep it running. And I think that helps. That background helps a little bit.
So nanoparticle industry is huge. It was introduced like publicly with COVID, with the vaccines and everything. But I feel like so many new things are going to be done in the future with nanoparticles. One of the coolest things I think is not organ on a chip, but a chip on an organ. So for example, like if you wanted to have some sort of blood glucose monitor where a level of glucose in your blood that is detected by this chip can release some sort of insulin or some sort of nanoparticles that help a diabetic. I think that's pretty cool. Nanoparticles aren't only just like a injection thing.
Yeah.
So like what do you think about the field of nanomedicine and outside of your use in the lab?
So I think in the last decade, it's kind of stagnated a little bit just because people are moving on to other things and trying to expand the field in other ways. Like CRISPR really changed the nature of like the biotech field in general. But now it's coming back kind of full force after COVID in terms of people being like, this can be combined in so many different ways with different devices and different technology. So I think there's still a lot of potential for nanomedicine applications, especially since it's so biocompatible. I just think that the research dollars haven't always been there in the last, from 2010 to 2020. Yeah.
I feel like that's the case for a lot of sciences.
Yeah.
I feel like they kind of get bottlenecked by what's like the new thing, and CRISPR is definitely one of those things. And it's great for science. It's great for, because I'm sure that there are ways to apply CRISPR in the research that you're doing, for gene editing and what you're packaging and how you want to maybe manipulate that DNA or RNA sequence. And I think that that's really, really cool. So,
does Johns Hopkins do drug delivery research too? They probably do, right?
Yeah, actually, so I worked in 2 different labs while I was there. Both of them were drug delivery heavy. One of them was doing cancer stuff, but looking at the physical microenvironment and how we can make one, understand the mechanical forces, and then do various treatments, including like having concentration gradients of specific nanoparticles. And then the other one was very much like drug delivery, nanomedicine, what you would consider conventional uses of nanomedicine through mucus layers, essentially.
Mm-hmm.
So, yeah.
Well,
That's
cool.
incredible. It's incredible. So,
It's exciting.
what what kind of is your day to day like in the lab? Because when we bring PhDs on here, it's always interesting to hear how everybody has like their different techniques and how they cope in the lab, and kind of the stress of the lab. And I like that. I like to talk about that because I remember as an undergrad, before I even got into a lab because of like COVID and everything, I was like just so like nervous about when I do step into the lab, like how I'm gonna look, how it's gonna change. So, like, how did you kind of your journey in labs? How did you learn everything and all that kind of stuff?
Yeah, I think when I first started research, my expectation was that I'll be shown what to do and then I'll be able to like replicate it perfectly, and all my experiments would be beautiful. But obviously, like that's not the case. There's so many setbacks and troubleshooting moments, and you have to kind of change your mindset to be like, okay, this is a learning process. We're developing new science. Nobody else has ever done it before, so just like be patient with yourself on a day-to-day basis. I went to a conference in California recently, and it was more focused on like professional development. It was Abercrombs, and basically somebody was presenting and saying like you should plan out your days so that 60% is planned and 40% is like unplanned, because 20% of that will be like unexpected things, like errors, whatever happens, and then the other 20% will likely be like spillover from like the day before or something. So
I like how they didn't say 50-50.
Yeah.
60-40, just to make sure you're in control.
Yeah. So I think that's a good rule to follow in terms of how to like break down your day-to-day in lab.
Yeah, I like that. I've never heard that before. But I think that can be applied to a lot more things other than the lab.
Yeah, in general.
It's nice
like,
good advice.
I do like I like that a lot.
Yeah.
How have you noticed your PhD evolving from the first year, second year, and now your fourth year, fifth year, right?
Yeah. Well, in your first couple of years, you're doing a lot of classes, and it's a lot of like figuring out a knowledge basis from which to work with for your projects in the future. And also you're asking a lot more, like, I would say, basic or quickly answered questions. And then once you get towards the end of it, it's a lot of like big head questions that, is going to take you like weeks to figure out, which I appreciate. That's one of the reasons of why I really like research, is like that ability to think about things. But yeah, the first, I'd say like the PhD is filled with moments that are sprints. It's not like, people like to think of it as a marathon, but there are like sprints throughout it where you'll be doing so much work and then you'll have time to like slow down, break it down, digest it, and then there'll be another sprint where you have to do a ton of work. Yeah.
That's really cool.
And life sciences is difficult because you got to know and read a lot, because there could be a paper out there that completely disproves everything you're doing.
Yeah.
So like knowing what you're doing first is important, and then going in that direction. So did Dr. Liu, when you first started your PhD, first let's unpack, how did you rotate under different labs here at Case?
No, Case is like a one-to-one match for BME.
Oh, really?
Yeah.
So meaning that you found Dr. Liu and that was it? Or
you interview with other PIs, but you like will rank your top, and the professors will also rank you, and then you match.
That's really cool.
And then what was I gonna ask? It was basically like your PhD, how when you started. Oh, Dr. Liu.
Yes.
You guys, how do you like working with Dr. Liu?
I actually like working with Dr. Liu a lot. He has a very particular personality, which is like all PIs, they're all very specific, even if they don't want to admit it. But generally, I think that he allows you to grow and like work with him. I spent a lot of like time like working one-on-one with him, like through big concepts and how we're going to figure out things. So I guess a little bit more background about my project. So we're doing molecular imaging of cancer, but everybody's doing it in a different way with a different cancer. My approach is with immunotherapies. I also do gene therapy, but right now the focus is immunotherapy. And our lab has never done immunotherapy before, but it's a huge part about figuring out how the tumor microenvironment works, which is what we're trying to image. So just like being able to talk to him like one-on-one about like, okay, I read this paper and I think that this is how this particular mechanism is working. And he'll be like, okay, I read another paper that says this is another thing that we should consider.
That's so nice.
So,
it's so like
collaborative.
Yeah, collaborative.
Yeah, and I think a lot of students don't necessarily have that opportunity, but I really appreciate that.
Definitely.
He knows that I want to go into academia and become faculty one day, so he's been able to like give me specific opportunities that are leaning towards like somebody who wants to go into academia, like working on grants with him, for instance. So
that was my next question is like, what's the next step once you get the PhD? You know, what would be the next step?
You're asking a scary question.
I know,
right?
It's so nerve-wracking.
Yeah.
Start paying my loans.
Well, the goal is to go into postdoc and then to faculty. I feel like very few people go straight from PhD to faculty.
Yeah.
Though that does exist. Yeah.
So when you first started, did you basically have an idea where you wanted, like, what disease you wanted to tackle with this PhD, or was Dr. Lu kind of like, hey, from what I've known, this would be a good avenue for you to go into, and then you emailed him back or something, and like, was it a lot of back and forth, or did he have a project in mind for you?
So we did go back and forth initially. I initially thought I was going to work more on like drug formulation, drug like delivery things, which I do now, but I didn't necessarily think I would be doing so much heavy imaging. So we're doing MRI, and MRI is very time intensive, and it's for every single part of my dissertation, which I didn't know any of the physics behind imaging, and now I'm like, oh my god, it's so cool. Like, very underrated field of study, so, but it's growing.
One of my things, like, so I don't know if, I'm a first year RGME student, like, I'm just now getting into, like, all the stem cells and everything, but one of my favorite things to learn about is the combination of regenerative medicine and technology, especially imaging and AI.
Yeah.
And I think that stuff is so cool. Combining, learning data from an AI system combined with a physician's, naked eye who is an expert in reading these tests, and then combining that knowledge to diagnose somebody. I think that is like
super comprehensive.
It's so cool. It's just so cool, because who would have ever thought 10 years ago or 15, 20 years ago that we'd be having, both doctors and AI reading. A lot of people are just like, computers are going to do everybody's job. And like, we're very quickly learning that computers are not perfect. We need the expert with the computer. And I think that is something that I'm very interested in, is the technology aspect of medicine and the technology aspect of research. Because without the technology, how much research could you really get done?
Yeah.
Right? So I think that hearing the combination of the MRI is something that a lot of people don't think about.
Mm-hmm.
You don't think about all the instrumentation that you use. Like, I use flow cytometry a lot.
Yeah.
How could we determine these cells in a compact amount of time that wouldn't take us forever and a ton of resources without a flow cytometry machine?
Yep.
And gets read. So technology is very, very important in research, and I like hearing that you guys use MRI for almost every assay, because that's important. You got to see what you're doing.
Yeah, and I think visualizing what's going on was like previously a little bit underrated, and now everyone's like, especially for clinical stuff, you want to see what's happening. So yeah, imaging has like huge potential to blow up.
It really does.
So.
I think one of the coolest things Victoria's lab is involved in is that, like, so some labs, they'll do nanoparticle research and gene therapy research, but they'll use an already out there lipid, or they'll use micelles or things like that.
Mm-hmm.
Dr. Lu's lab has developed its own nanoparticle, which they have patents on. When I break that down and think about it, I'm just like, how cool is that? You can say you have your own nanoparticle. There are people that, like, I've talked to professors here that they say, oh yeah, I do nanoparticle research too, but they use micelles and things like that are not theirs. Everybody in this lab, like, I think, Victoria, you have your name on the patent as well?
I actually don't. I have my name on a different patent, but
Okay.
it's related to
Yeah, but
ECO.
you're probably like a year or two off
Yeah,
from like
yeah.
actually getting your name on it, because like a couple years before her is when like it got established.
Mm-hmm.
So like Ryan and all of them and whoever his grade area was. So that I think is the coolest thing about Dr. Liu, and especially the whole imaging thing. He has his own developed his own MRI contrast agents too for for cancers.
Yeah.
That's super cool.
Yeah.
Yeah, I think so. I mean, I like it, but I have mixed feelings about creating new technology versus relying on like old technology and improving it, because you have to find like a really nice balance between the two, where it's like economical and efficient to use like the new technology, and then also not necessarily like usurping old technology, because it's really hard to translate like something that is completely different from what anybody has ever seen.
Yeah.
Yeah. So.
So are you saying it's harder to make new technology sometimes, or it's harder to translate old ones into new ideas?
I'm saying that it's it can be difficult to integrate new technologies into like current like practice, for instance.
Because you already have so much data
From the old stuff?
Because you guys have been using
Yeah.
MRI your lab has been using MRI for how long? Imagine if Dr. Liu came in and he's like, I bought this brand new machine, it does all this stuff, and you're like, well, now we have to take everything we just did and translate it to here. We don't even know what this thing looks like yet. So,
yeah.
I don't know.
So I think like for instance with AI, people like it a lot because like you're using the same imaging modalities. The only thing is that you're training like, machine learning neural network to figure out various patterns. And that you don't have to necessarily like get rid of all of like the imaging tools that are used in hospitals today, but you add on like substantial knowledge to what is already going on. And it really just depends on where in the field you are.
Outside of nanoparticles, what do you think is the coolest new medical technology? Do you believe in Neuralink? Because over the summer we did talk about it in the lab a little bit.
Yeah.
Do you like prosthetics? Do you think, I mean, they obviously help people a lot, but in terms of coolness, what do you think is like the coolest thing?
Oh, this is really hard because there's so much in the scientific field. I guess because I also work with immunotherapies, my bias is going to be like, yeah, immunotherapy. CAR T cells are like,
awesome.
The therapy is really cool. And just in general, I think a lot of the immunotherapies are really exciting. They're mainly using antibodies or like, it's bolstering your own immune system, which I think is like very exciting.
Yeah, just got to make sure we keep it under control.
Yeah.
That's the one big problem I think they're having right now. If I had to pick one, and just like science in general, I think that the deep, like this is so literally off the deep end. I think that the deep ocean is so cool.
Yeah.
I want to go study the Mariana Trench. Like if I wasn't doing regenerative medicine, I might be a marine biologist because I think that stuff is so cool. Isn't that? I understand.
It is cool,
but it also opens doors for like new drug discovery because you can go down there and find new molecules that are made from these sulfur-eating bacteria that have never really seen oxygen before, and like they have some different enzyme that we could potentially use.
Yeah, specifically with the Mariana Trench, they found like all these like ore that are like at the bottom of the trench, and they want to use it because it has like lithium, cobalt, so basically anything that you need for like an e-vehicle. Yeah, to like make the batteries and all of the circuits. So they're looking into like not mining it per se, you're like literally raking it up and like taking it from the bottom of the ocean, but nobody knows about the implications of what's going to happen to like
you said
deep
these are
sea.
Oysters?
The ecosystem.
No, like ores. Like
ores?
Yeah, like ore.
Yeah.
Like little
the
balls
rocks.
of like
Rocks.
Yeah. Of like metal.
Yeah.
Yeah.
Mariana
It's
Trench
very exciting.
The only reason I say that is because I saw a picture of some translucent octopus that they took a picture of, and it was like, that's really cool. Like you can see its brain and then like all of the nerves running down its tentacles, and you're like, who knows what else is down there?
Yeah.
You know how turmeric is anti-inflammatory?
Mm-hmm.
Some scientists have tried taking turmeric extracts and using it for cancer therapy to make it anti-inflammatory. Really?
Yeah. So I was at this big consortium called the Center for Nanomedicine. It's where like our main PI, he used to work under Langer, and so he like founded it. And one part of like our lab was doing curcumin extracts for colon cancer. and turning it into nanoparticles for better absorption.
That's cool.
I hate that name though, curcumin. Cumin is another thing.
Yeah, yeah.
So curcumin has to do with turmeric. I'm just like, what?
Yeah, it's
Like
confusing.
Scientists, they got to make it easier.
The
A little
naming.
bit
Yeah.
easier.
I still
Yeah.
love Sonic Hedgehog.
Oh yeah, there's some disease, right?
It's like a receptor. It's a protein, right?
They call
Yeah.
it Sonic the Hedgehog.
Yeah, it's
Yeah.
called
I don't know why.
SHH, Sonic
I
Hedgehog,
forget why.
That's awesome.
because that's what they named it.
Yeah.
Isn't there a reason though? Like,
Does it look like it? Does it either moves really fast, collects gold coins, or has
Hedgehogs.
really long hair?
Yeah.
Looks like a hedgehog. It might actually. It probably looks like a hedgehog because of how the 3D proteins look sometimes. But
The
Did
various
you-
conformations.
did you
yeah,
ever
watch-
possibly.
Did you ever watch Star Trek?
Yeah.
You did?
Mhm.
Okay, I never did. But anytime I'd talk to somebody about regenerative medicine or something, they're like, oh yeah, have you watched Star Trek? Because apparently in Star Trek, there's like these scenes where if you had like something wrong with you,
Uh-huh.
you put it under a machine, and this machine would just like grow everything back. And I think like that level of sci-fi is really cool to bring people interested in medicine.
mhm,
But when we try translating things, it becomes less cool.
it's really hard.
Yeah.
It's
Well,
really hard.
Yeah, you have to like balance like actually giving appropriate patient care, so you don't want it to like be 3D printing somebody's arm back and then it's like not their arm
Oh,
or
okay.
like those cells aren't, they don't differentiate.
Yeah.
But yeah, we saw a very cool speaker on Thursday. He was working with ECM like sheets and using it to like grow back like muscle tissues for veterans basically.
Oh, that's really cool.
Yeah, and it was pretty effective. So all you need is some ECM sheets apparently.
Over the summer, I showed you this one thing created by Dr. Kaplan. He's like the father of stem cells here.
Mhm.
He uses placental tissue that's just medical waste after giving birth. And he uses that placental tissue and mixes in growth factors and ECM
Mhm.
to create this membrane in this product that can be literally put as a band-aid for anything. So like for diabetic foot ulcers, somebody will literally have degeneration of their entire like foot, and you'll see them literally start to have necrosis and just slowly corrode. And all you do is put these stem cells, and because they're come from placental tissue, there's no real graft versus host disease that could even happen. You put this tissue on there, and it literally grows back everything that's missing from your bone to your fat, everything. And it's like
I wish my brother had
a
that.
fully reimbursed by Medicare product created
Wow.
by somebody here at Case,
That's really
and it's
cool.
just like the coolest thing ever.
My brother had a really bad staph infection on his leg. And it got ugly, but they basically had to take out his whole like shin, not like the bone, but down to the bone. And they did a skin graft, and like it looks pretty good. Like the plastic surgeon did great, but that would
Well, they're not stem cells though, right? Like
they were his own. It was his own skin that just kind of grew back. It still isn't technically grown
technically,
back, but
yeah.
it's been like a year now.
Wow.
He's probably got another year. But if that placental Band-Aid was there, I'm sure my parents would have loved to pay for that. That's
It's
really
covered by Medicare though, so it's just like your insurance would cover. But it's just like
That's really cool. Like that's so cool. Like I wish more people knew about that.
Yeah.
Because how many times do people get skin grafts, for burns or anything like that, and they're taking skin from their back, from their calf, and
Or like even like the porcine ones, there's some people who have like ethical reasons why they wouldn't want necessarily like a
Yeah.
pork graft,
Yeah.
and that would be nice too.
or like
The pig heart?
pig heart, pig
Yeah.
aorta.
But
Or
that poor
for
guy.
children too, who don't have like a lot of tissue to take
Mhm.
from like a different area, and you have to like graft it.
Like my brother was 15,
Yeah.
and he didn't have a ton. He didn't have like a ton of skin, but I mean they did a muscle and a skin graft, and it was wild.
Well, let's see. Okay, if everyone knows about it, you're gonna run out of inventory of the placenta tissue in like 5 seconds.
Okay, fine.
So people gotta donate, and people gotta be able certified to apply it. So the doctors, surgeons can't. I'm not sure how the certification works. Do you have to be certified to do some stem cell stuff, or if you're a surgeon, you can do whatever?
No, I think the technology has to be like, verified and
Verified,
yeah, in terms
okay.
of like using it in like the
But
clinic.
But Kaplan has this thing on the market already.
Dude, since 2014.
Holy crap!
Wow, that's
Yeah.
crazy.
That's so cool.
but
Yeah.
nobody knows about it because
That's why
nobody
we-
knows about stem cells.
That's why we do this.
Yeah,
so
that's
that
what we're doing.
the people-
I think the thing is like stem cells are always like in flux in terms of like how people feel about it because you know for whatever like for instance for the abortion ban the things like that people are always like the next step is like banning stem cells
Oh.
because one of the reasons that like stem cell technology came like about was because it was for IVF like treatments
Yeah.
and that's how they started researching more stem cell technology.
Yeah, but now we don't even use embryonic stem cells a lot.
Yeah.
Very much. It's all hematopoietic stem cells,
Mhm.
induced pluripotent stem cells, which is still brand new, mesenchymal stem cells or mesenchymal stem cells, however people say it up here.
Yeah.
Mesenchymal.
And there's so much more. I remember coming, from Cincinnati, which, like I said earlier, is a very Germanic Italian Catholic area.
Yeah.
I was telling people, I was like, I'm going to study regenerative medicine. And like, what is that? I'm like, it's stem cells. And they were like,
Devil.
oh my God.
Yeah.
I was like, you don't just let me do it. It's fine. I'm not, I promise I'm not killing babies. Like, that's not what we're doing up here in the labs. Like, and people were like so just besides themselves about it. And I was like, you don't even understand the half of it. We're going to go up there. We're going to save people's lives
Yeah.
because of this. So, yeah, I think that it does have like a bad stigma for like no reason. It really does. But hey, we're changing lives. Arnold Kaplan already probably changed a bunch of lives. So, and nanomedicine is going to do it too. So
I think the two go hand in hand. I was thinking about this the other day and how basically most of the diseases we're fighting in terms of like regenerative medicine or like with cancer are things that are related to aging.
yeah.
Like realistically, like the biggest factor is how we age and how our cells respond as we get older. And that is like, I don't know, the connection between everything.
Yeah.
It's how everything's enmeshed.
Yeah, age and time. We're fighting against time.
Yeah.
Is what we're doing.
Do you guys know about this group? There's like, in the past 100 years, these really, really rich people, they'll like pay hundreds of millions or X amount of money to this group company that'll freeze them. So there's like dead people from like 100 years ago
Yes, I have
that are
heard
supposed
that.
to be dead 100 years ago that have been frozen still. So my question to you is, when do you think those people are gonna be back?
Never.
Oh.
Never.
Impossible.
I mean, I don't know if we could like, you know, pull a zombie type of situation and like reanimate people.
Yeah.
I'm sure somebody could come up with a way of doing it, but I think the ethical implications would be hard to like.
You know that Dolly the sheep
Yeah.
that was cloned?
Mhm.
Could you thaw these folks and then just
Take their
somatic cell nuclear transfer them and clone them? Because
Maybe.
technically those old cells are dying. I don't know how that works.
I think
don't
it's
know.
not like the time part that I think people get in trouble with freezing for a long period of time. It's not the time. It's literally freezing yourself kills you.
I mean, I don't know.
I mean, they were already dead. Like,
Right, they're already dead. Like, so, like, think about it. The cytoplasm is what, like 70% water?
Yeah.
What does water do when it freezes? It expands.
Yes.
And then it breaks. What it's going to do is it's going to
All the sheer force.
Yeah, it's just going to break everything, and then you're just going to leak out. And
Okay,
then
well, the woolly mammoths in the Arctic are fine.
they're built for that. They're not getting frozen for 100 years.
Nature froze them, and they're doing okay.
They're perfectly intact, even their
Yeah.
stomachs.
Yeah, really?
Yeah, their
Really?
stomach contents are like
But are
perfectly
they
intact.
walking?
But why do nanoparticles exist? Because somebody made them. These are natural things.
Yeah. Okay. I mean, maybe I'll be frozen in 50 years, and you can laugh at me.
I mean, I'm sure there's a window of viability, but
True.
yeah, I think once you like mess with the water content, since that has to do a lot with our like electrical signals throughout the body, and that's really what you would need to like fully reanimate a person. I mean, along with other things, but like if you can't get the electrical part going, then your whole nervous system isn't going to work.
Walt Disney's not coming back.
Is he frozen?
Yes.
He's frozen?
Yeah.
No way.
Kind of don't want him to come back.
No, he doesn't need to come back, and he's never going to.
You don't know that.
He's not.
I feel
And
like
how
he
were the
would,
first-
he would get real stressed about
Okay,
watching
all right, we are
content.
getting off track. So, man, we're at 35 minutes already.
I know that was nice.
That was quick. That felt
Good
quick.
combo.
So, well, it was great to have you on. It was great to hear about your
Thank
PhD.
you.
It was great to it's nice to talk about your PhD, but it's also just nice to talk and learn about people and the interests and even opinions on freezing people. So I think that was good today. So do you have anything you just want to say to maybe anybody watching? Maybe something you would say to yourself 5-10 years ago that would help you like maybe prepare for a master's or a PhD?
Ooh, yeah. I think one of the things you brought up was like how your research thought process has changed over time. And I think always ask questions, like no matter what stage you're in your PhD. It's just really important to be like an inquisitive like learner. And you'll get a lot from that, like from the people around you. Ask questions from experts in the field, and you'll do well. Yeah.
Perfect. All right.
Now, one last thing? How do you find all these conferences you go to? So like, are you on Google like Biotech Conference 2022?
Yeah. So like, journals will send you like advertisements because the journals are the ones who are like sponsoring the conferences.
Oh, okay.
Yeah, and you can just ask people in your field like, "Hey, what's the big stem cell conference?" Or like,
You know that one?
Yeah.
That one time, and they're like, yeah.
Yeah, professors know of a ton of them, and they're happy to like share it with you because any journal they've published with will likely send you like some advertisement about their conference. So yeah.
Cool.
All right. Well, I think that does it. So we want to thank you for coming on.
Thanks
It was
for
a pleasure.
having me.
We'll
It was
definitely have
fun.
you back.
Good luck on your
if you want to-
PhD.
Yes, good luck on your PhD.
Thank you.
And we just want to say thank you guys for watching. This has been Cells to Cells. See you guys in another one.
See ya.
Bye.